I was at home during this week.
You already know most of you Ricardo. Ricardo Martínez is he finished his PhD 4 years ago, more than 4 years ago, here at the FIS. Since then, he's a postdoc researcher in the Department of Ecology and Evolutionary Biology in Princeton, and today he will be here at the FIS for 1 week.
Until Wednesday. Well, Monday, Tuesday, and Wednesday.
It's in the office 103, I think. Okay, well, he will talk today about the collective behavior in microbial populations.
Thank you. Well, thank you, Cristobal, for the introduction. Thank you for coming, and Happy New Year, first of all. So the work I'm going to present to you today is basically a summary of things we've been doing for the last 4 years at Princeton, and actually tries to discuss how, by looking into behavioral processes, or by looking into short-term processes, we can at least theoretically try to infer ecological and evolutionary processes. So how can we go from information that we obtain by looking into behaviors to go to ecological and evolutionary scales? So if we understand collective behaviors in a very broad sense, basically by any system in which we have a lot of individuals that somehow are coordinating or synchronizing their behaviors, then we find a lot of examples of collective behaviors in nature. For instance, probably the most famous ones are these flocks of starlings, all these birds flying all together in coordinated ways. We also have these swarms in locust, where these insects transition from one phenotype to other depending on the environmental conditions. But we do also find these collective behaviors in the smallest scales. And actually we have, in microbial systems, we have many cases in which cells coordinate with each other to perform some collective task. And even if we move beyond these processes in which basically the synchronized behavior is movement, we also have this synchronization affecting other kinds of traits. So for instance, we have reproduction can also be synchronized. We have this example in bamboo, in which basically a lot of plants flower in a very short time period. And we also have these wildebeest, in which they are also famous for these huge migrations, in which they all migrate together from one place to the other, but they do also give birth to their babies in a very short time period during the year. But if we also go to the smallest scales, we do have microbial collective behaviors also affecting different traits beyond movement. We have probably the most well-known example are bacterial biofilms, in which we have a lot of cells crowding all together, and somehow they coordinate collective behaviors that are basically reflected by the secretion of molecules to the external medium. So we have a lot of examples of systems in which individuals somehow coordinate their behaviors to do some task that they could not do by themselves. And in particular, all these behaviors are very different between them, of course, but they share some features, they share some properties. And today, the one I'm going to discuss is what's the degree of synchronization that we observe in these systems, and why sometimes this is not perfect. And there are also, among the systems I was introducing, we have, for instance, these swarms of locusts, in which, as I was saying, basically these guys live in a very solitary state, but when they are exposed to a very crowded environment, in which they have a lot of insects around them, they transition from being solitary and repelling from each other to being gregarious and attracting from each other. But there are experimental results showing that even if we expose these insects to very long times in very crowded environments, not all the insects will transition from one state to the other. There is always a probability, not equal to 0, to find solitary phenotypes within a population. Another example is, as I was showing, this calving season in wildebeest, in which we do have most of the calves being born in some time of the year, but we also do have some tails in this distribution. So in this context, in which we have all these collective behaviors, and we have some of them that we know that they are not perfectly synchronized, we there's a question that is, at least for us, very appealing. So what is the question here? Going to the most famous sentence in this institute. I had a picture here that I'm not going to show, but the question is this imperfect synchronization just an unavoidable byproduct of this larger scale synchronization process, or is this something meaningful biologically? And if it is something meaningful, then the question is, what are the causes of this imperfect synchronization? What is driving these non-perfect synchronization processes? And what are the consequences? First, if it has some consequences, and if so, what are those? So to address these questions, we are going to use as a model system the slime mode Dictyostelium discoideum, which has been a very well-known model system for multicellular development and microbial social behavior. So the work I'm going to present has been done mostly with Fernando Rosine, who did almost all the experiments. He's a grad student at Princeton. Also with Alison Gross, she's an assistant professor at Boston University now. And with Thomas Gregor, who is a full professor at Princeton, and with Corina Tarnita, who is an associate professor at Princeton. So, as I was saying, Dictyostelium discoideum is a very well-known model organism to study these collective behaviors in microbes. And the reason why it is because it has a life cycle that actually combines a unicellular phase and a multicellular phase. So basically, in the presence of nutrients, these amoebae, this is an eukaryotic organism. These amoebae feed on bacteria, and they divide in a unicellular phase. But once they consume all the resources in the environment, they start signaling with each other. They start coordinating with each other. And as you can see in this movie, they will eventually migrate all together to the same place in the system. This is assumed on some region of the plate. If you go to a larger scale, there are actually many of these aggregation territories close to each other. So once we have this multicellular aggregate form, then this clump of cells will start a developmental process that will lead to the formation of this fruiting body that you can see sketched here. So basically, some cells will become reproductive spores. They will be raised to the top, and they will be dispersed in the environment. And some other cells will die to form this stock here. But before all this happens, so actually at the end of this aggregation process, in which I'm stopping the movie here, there is something that has, that at least I want you to pay attention to. And it's the fact that in addition to this multicellular clump, we do have some cells that did not follow the multicellular process, the collective behavior. So it's more clearly shown in this larger picture here. I'm just highlighting some of them, but I think it's quite clear that there are some cells that just did not respond to this to this collective behavior. They are solitary, and they are there. So, of course, these cells will have different properties than these ones here. So these ones will be more resistant to the absence of resources. They will survive longer times. But these ones, even though they are less resistant, they will avoid all this developmental process. And if the food goes back to the system quickly enough, they will be able to start dividing and start exploiting those resources much more quickly. So we do have some intuition. We know that these cells are there, but we do not know what's happening with them. We don't know if they are noise, if this is controlled somehow by the collective process. And that's actually the thing that we need to quantify. So first of all, we need to show that these cells will be alive, basically, that they are not dead there. And in order to do that, we did a series of experiments. Actually, Alison did these ones, in which we basically plated some amoebae. We gave them bacteria. And after a day, they consume all the resources and they aggregate it. Then, once we have these fruiting bodies formed, what we do is we remove the spores, which we know they will germinate somehow, and we basically only leave in the system the stock cells that we know that they are dead, and we also leave these solitary cells that did not aggregate. At this stage, if we give bacteria again to the system, to these cells, if they start dividing, then this means that these solitary cells will be alive. So we recover the nutrients in the environment, and what we observe is that after some time, the cells that did not aggregate, they will start consuming those bacteria, they will be starving again, and they will recover the multicellular phase. So in principle, for at least some time, these cells will be surviving starvation periods. So with this intuition, the first thing that we did is a very simple model to see whether this may have some ecological implication for the system. So for some ecological implication for this organism. So starting from very simple hypothesis, basically what we assume is that once these guys don't have any food, then a fraction alpha of the cells will aggregate and a fraction 1 minus alpha will not. So basically we have two phenotypes with the different properties that we know. Ones will die very quickly, the other ones will not. And we assume that this partitioning behavior is controlled by the organism. So we assume that each genetic variant of this organism has a fixed alpha, has a fixed partitioning behavior. So with these results, we can basically label each strain, each genetic variant, by how many cells become part of the multicellular aggregate. And we can explore how different environmental conditions, that here I'm representing by how long food needs to recover, how those different environmental conditions select for different partitioning behaviors, basically. And when we do that, we find that, as expected, as the environmental conditions become worse, then strains that invest more in the aggregated phase will be selected for. So basically, because these cells are more resistant to starvation and they will live longer, if the starvation periods are longer, they are selected for. In this other extreme, we have selection for strains that do not invest in aggregation, basically because they will be very ready to start exploiting the nutrients. And in the middle, because the environmental conditions, I forgot to say, are stochastic, so basically this is the mean starvation period, but we have some noise around this mean starvation period, then it kind of makes sense to put a little bit into the aggregated phase and a little bit into the non-aggregated phase just to avoid these fluctuations.
Is this experiment or model?
Sorry?
Is this experiment?
Sorry, this is a model. This is a very simple model starting with only this hypothesis, and basically one population dies with some rate, the other one dies with longer.
And alpha is what you call the genetic variant?
Alpha is basically the proportion of cells that go into the...
Is the vertical axis in the plot?
Yes. Yeah, I didn't put it, yeah. And this is frequency, basically. So, at least theoretically, we predict that there are some selective benefits for these cells that don't aggregate. So, at least these models are telling us that in given environmental conditions, this non-aggregated phase can be selected against the aggregated phase. And in addition to that, if we couple this simple model to environmental heterogeneity, basically we run this model in different patches, in a network of patches with different environmental conditions, and we couple all of them through dispersal, we observe that as dispersal increases, then the diversity of strains coexisting increases as well. So basically, the fact that we have different patches selecting for one single genetic variant and then dispersal mixing all these patches kind of increases the diversity in the system. So the take-home message from these results are, take-home messages are actually two. The first one is that investing in non-aggregation may be favorable in some environmental conditions, and when coupled to environmentally heterogeneous conditions, that may explain biodiversity patterns in these species. And one can also think on different types of environmental heterogeneity. So for instance, instead of having different islands with different conditions, we may have one single island with seasons, for instance, and in those cases, we do also have coexistence of species. So these results are telling us that these non-aggregated cells could be meaningful, right? They could explain biodiversity patterns in this species, and they could be playing a very relevant ecological role in this organism. But as I was saying in the beginning, this model departs from two hypotheses. The first one is this partitioning behavior. So basically, in every time that these cells starve, a fraction alpha become aggregated cells, and a fraction 1 minus alpha becomes non-aggregated cells. And then we are also assuming, and that's a very strong assumption, that in each starvation period, that alpha is fixed, is constant. And that's something that we really don't know. I mean, we don't have empirical evidence of these processes being something that is heritable. We don't know if through different generations that's conserved or if it is not. And if we want to validate the hypothesis of these models, we need to go to the lab and we need to measure those assumptions and see whether they are true or not. And that's the second step that we did. That's actually something that Fernando did in the lab, in which he was basically counting the number of cells that did not aggregate. These are experiments. So density of cells that do not aggregate, counting the cells in the plate, as a function of how many cells there were initially in the system. And when looking into this data, what we found is that when the initial cell density is very low, then aggregation doesn't even start, and therefore all cells are loners. So we need some quorum to start the aggregation. That's something that was already known, so nothing very surprising here. However, once the plated cell density goes above some threshold, what we observe is that the density of cells that don't aggregate is independent of how many cells we had in the system initially. And if we repeat this experiment for different genetic variants, we find that different genetic variants have different investments in non-aggregated cells. So basically, the density of cells that is left behind by different genetic variants is different. But because it is different, and it is a well-defined quantity for each strain, this is showing us that actually one of our hypotheses, the fact that this is a heritable quantity, is correct. But what it's telling us is that our hypothesis about partitioning behaviors is not correct, right? If we, given the assumption that we were taking of this fraction alpha being of one phenotype and 1 minus alpha being of another phenotype, one should expect this to be a line, right? A straight line with some given slope, and the slope should be alpha, right? Yep. But because we don't have that, then we are partially showing our assumptions to be correct, but we need to reformulate the way we are looking into this collective behavior. We need a new model to explain this lack of synchronization, basically. So that model is actually going to be extremely important to be able to go back to the question that I was asking in the beginning. So first to understand what are the mechanistic causes of those loners. Where are they coming from, basically? And then whether they have some ecological consequence. Those are the two questions that I will focus for the rest of the presentation. So what are the mechanistic causes of these loners? Well, what we propose is that these loners are the result of this aggregation process being stochastic, so having cells responding to aggregation with some probability, and that probability being a function of signaling, of cell signaling, right? That's with words. With figures, I'm not going to put equations. With figures, what this means, I have to say that what we are doing here is a random walk-based model. So basically, it's an individual-based model in which we have every cell moving with different rules depending on whether they are aggregating or not. Basically, if they are not aggregating, then they don't move, and if they are aggregating, they move. It's not super sophisticated model. It's actually a very simple model. So what I'm saying with this proposal is that we assume that initially all the cells are in the same identical state. We do not have any heterogeneity in cell states. And that's a strong assumption. One may think that they have different starvation, different energetic reservoirs or different conditions. We are not assuming anything of that. All the cells are identical in the beginning. And they are all signaling with some intensity, and therefore there is some concentration of signals there. And because this population is very crowded, we have a lot of cells, then the concentration of signals is very high. So in the figure that I was showing before, we are above the threshold. We are in the phase where the aggregation happens. But because this aggregation starts stochastically, what we do have is that only some cells, that here are the yellow ones, will start moving towards the aggregation center. The other ones will not. And that's something given with some probability. And then as these cells start moving and start migrating towards the aggregation center, what we find, what happens, is that the local density of cells decreases in some parts of the space, and therefore there will be some regions where the concentration of signals will be low, and cells will be frozen in those positions without the possibility of becoming aggregating cells, basically. And as this process continues to happen and continues to happen, then at the end, what we have is that there are some cells that become aggregated cells and some cells that don't. And actually those cells, which are going to be the loners, are basically cells that at some point they could make this transition, but they didn't make it on time, basically, because of this noise coupled with the quorum to start aggregation.
I have a question. I need to point. The cells don't, but look, why there is an aggregating point somewhere? What are the characteristics of that point?
So basically, in order to be able, because at some point, in order to be able to solve this model analytically, we assume that we have some space and we have an aggregation center that is fixed, imposed externally. But we know from the experiments that this is not correct. This is a self-organized process, and this aggregation point emerged basically from the collapse between signals and heterogeneities. So this is the assumption that we take in this model to be able to get some analytical results, but we also test without imposing this, and the results are the same. So in principle, what we are modeling here is only one aggregation territory, so basically the movie that I was showing in the very beginning, where we have only cells going to one point. But we do also check the fact of having a larger system with a lot of cells, and then without imposing any aggregation territory, and just due to the diffusion well, basically due to the dynamics of the signal, at some point you have this population to start aggregating in territories, basically. And for the movement, we only, what we assume, again, is that any cell that becomes the transition starts moving with some constant velocity in straight lines. We don't have any like imperfect chemotaxis or things like that, straight movement. So with this model, of course, the fraction of, the number of cells that belong to each of these populations is going to depend on how strong they signal, on how sensitive they are, and on the parameters of the model, basically. But what we recover is that the numerical, the simulations from this, the simulation from this model recover the two phases. So, of course, when the initial population density is very low, then aggregation doesn't start, and we have this one-to-one correspondence between non-aggregated cells and cells in the system, and then we have this plateau. So basically, what this model and these experiments are telling us is that we can classify different genetic variants depending on their aggregation performance. So for instance, we may have some, let's say, green strain that aggregates very badly, then we have an orange one that is the best one that leaves fewer loners, and we can rank these different genetic variants depending on how much they invest in one population versus the other. So now we know, at this point we kind of have the answer for the first question. We know that this population partitioning could be at least according to our proposal, that's only one proposal. Of course, you may come up with many different mechanisms, but at least from our proposal, this is the result of this stochasticity in the aggregation process coupled with some quorum for aggregation to start. So we do know that these cells are somehow regulated by different strains, and we do know that they are heritable and that they could be something meaningful. So then the next question is, what are the ecological consequences of this behavior? How is this behavior affecting the patterns of biodiversity that we observe in this organism? And actually, this is a very interesting question, at least especially for this organism, because slug modes are super diverse. Even in very small scales, you have a lot of species coexisting with each other. And actually, when two different strains happen to be in the same place and happen to starve in the same place, they don't discriminate each other. So basically, if we have these red cells and these blue cells together, they will mix and they will create this purple fruiting body. They don't separate, and they create these genetically heterogeneous structures. So then the question is how these interactions happening there that we can quantify by measuring these collective behaviors may explain or may have some ecological implication. So the first thing that we need is to, because I was saying that they mix together, the first thing that we did experimentally is to see how these interactions are changing the behavior of the strains. And to do that, what we do experimentally is to basically mix what I'm calling blue cells and red cells in different proportions, allow them to aggregate, and then counting the number of loners, and see whether they deviate from the expectation that we would expect if they don't interact. And I mean, first of all, if they deviate, and then how much, basically. So in this, what we obtain experimentally is that actually, depending on the frequency of each strain in the mix, we do have deviations from the null hypothesis. So basically, there are some developmental interactions due to cross-signaling between strains that are making these loners to be different from what we should expect if they don't interact at all. So once we have this experimental result, we can go back to the model and see whether we are also capturing this with our model, basically. And when we do that, we find that, the model does not only recover this kind of sigmoid interactions, but because with the model we can explore a much broader parameter space, what we find is that in this much larger parameter space, we do not only have these sigmoid interactions, but we also predict the possible existence of different patterns of interaction between these two strains. But let's focus only on the one that at least we found experimentally for this pair. So this is the result from numerical simulations for a kind of sigmoid interaction. The model has here a very strong thing to provide us, and it's the fact that we can know from this total population of loners how many cells belong to one strain and how many cells belong to the other one. And that's something that we cannot do, at least for now, experimentally, because making these cells fluorescent, at least the natural strains that we are working with, is very hard. So there are ways of making them, like in experiments, to make some cells green and other cells not green, and then counting them. But to do that, you have to modify their behavior in a way that will affect these results as well. So to have access to that information, we need this model and actually see how many of these cells the model predicts to be of one strain and how many predicts to belong to the other strain.
Just to make sure I didn't misunderstand something.
Yeah.
You said that cross-signaling could be one reason why you get spread
Mm-hmm.
in the distribution. But particularly for 0 and for 1, you have very large spreads of the distribution functions. So this cannot be cross-signaling.
No, sorry. This so for the distributions is actually for the extremes is natural variation. So when we repeat, this is for this is basically the same experiment that I was showing before, but 1 point, basically one initial density, the one that I was showing in the plateau. And basically, when you recover the distribution from these experiments and from the other ones, you obtain the same distribution. So we think that's natural variation in the behavior of the strains. So basically, this is an stochastic quantity. What I'm saying is, could be cross-signaling is the mean deviation. And this is something that I know, so this result was actually a very tricky statistical test, because first thing you think is, well, here I put error bars and this can be a linear regression. So we did 4 different statistical tests, like model selection here for different parameters. We also did like bootstrapping of this distribution, and we obtained like this is, a very strong evidence of this being nonlinear, basically. But yeah, so the variance here we think is natural. Well, at least in the extremes, if natural is natural variation, it's more about the mean deviating from the linear regression. But yeah, this was probably the most tricky part, showing that this is non-linear. So yeah, going back to this point, when we look into the composition of the populations, what we observe is that the model is predicting that due to these interactions, one of the strains in the mix aggregates even worse than what they did without a partner, and the other one aggregates even better. So the straight line here is what one should expect if there is no interactions, basically, right? Because we do have, in the lack of interactions, we have the null hypothesis here that should be composed by the same fraction of each strain, but we observe strong deviations from that null hypothesis. So going back to our aggregation performance space, what we observe is that due to this cross-signaling, due to this mixing together, aggregating together, strains diverge in their aggregation performance. So basically, when these two aggregate together, they behave even more differently than what they did when they were alone. And we can measure that by looking into the behavior of the strains. So we can see whether by interacting between them, they change their behaviors, and now we can at least theoretically see whether these changes in the behavior have some implication in the longer scale, in this case, in the ecological scale. So going back to what is the question here, the question here is what are the ecological consequences of this self-organized behavior? And to do that, basically, what I'm going to show you is how the model predicts the ecological dynamics to happen as we have several aggregation processes one after the other. So basically we have competition for resources between two strains over a very long time, and we are going to see which one outcompetes each one. So let's also compare for these dynamics, the effect of aggregating in segregated scenarios, so basically not mixing, versus aggregating in a well-mixed case. So if we start by segregated aggregation, basically what I'm showing here is 25 different patches with the same ecological conditions. They are all identical and they are not linked with each other. They are independent. So basically we have competition between pairs of strains in each of these squares. Each square is identical. We start with the same identical conditions, and the only different thing that we have is the stochasticity in the environmental conditions, basically. And because this is a segregated aggregation, so basically the red one is the orange one, if you want to say, is aggregating to the left, and the green one to the right. They are not mixing with each other. What we observe is that as this competition happens, one starts outcompeting the other, displacing the other from the patch, and then at the end, the green one wins all over the place. So remember that here, these two strains behave differently, but not so differently. They were kind of in the middle, both of them, if you want to go back to this aggregation performance space. But what happens when we go to the mixed case, so to the case in which we actually have that curve of interactions affecting the aggregation behavior of the strains? Well, what happens is that initially, I said in the beginning that the initial condition was the same. We start with different frequencies of each strain in each patch, as you can see from here. Right? So basically the fraction of cells belonging to each strain is different across patches. And in this case it's the same, but they are mixed. So when we run these dynamics through different aggregation processes in these different patches, what we observe is that due to these developmental interactions, the transient dynamics is much shorter, but then the uncertainty in the winner is also bigger. So basically, the competition time is shorter. In this case, in the case of well-mixed aggregation, there is always one strain that is going to eliminate the other, and is going to do that in a very short time, but I cannot really know which one is going to eliminate the other one. Whereas in this case, I know with complete certainty that the green one is going to win everywhere. So basically what this result is telling us is that due to these developmental interactions that we are measuring by how we are measuring through changes in the behavior, then the diversity of the species can be affected at different scales. So basically we are looking in the lab into how the behavior of the green and the orange strain is changing due to these interactions. And from those changes in the behavior, we can predict, we can try to infer changes in the ecological processes that are happening on a much larger scale. And going back to kind of trying to put this on a broader context, we started from a very particular observation for this model organism. So we observed that there are some cells that do not aggregate in these slime modes. Then by looking into how this self-organized process happens, by what are the mechanistic rules that are driving this behavior, we can try to infer important effects of this asynchronized self-organized process into much larger ecological scales. And with this, if we try to put this on a broader context, what we can say, the way we can look into these self-organized patterns is by them providing us a link to extrapolate or to translate information that we are obtaining from individual level processes, from individual behaviors, to system level processes that can go from ecological and even to evolutionary scales. And just to finish with very future directions that could go from this work, the first one that I think is very interesting is to try to put these collective behaviors in the context of social behaviors in microbes, actual development of multicellular structures and how those could be affected by different strains that actually do not participate in these collective behaviors, and then try to see whether this imperfect synchronization is providing us with some way of making these systems more robust against these free riders individuals. And with this, well, thank you for listening. Also, these are all the people working on the project. Alex Washburn, he's now at Duke, and Simon Levine were involved in the first part of the project. And these are the references and the well, funding, of course. Se está estudiando.
Well, in the first part, when you develop the model to explain that the donors become independent of the number, in your model you get a very nice flat response, but in the experiment, the average might be like that, but if you slightly change the size, the fluctuation were huge. Not here.
If we have fluctuations there, then we'll have a problem.
Well, here for you.
Yeah. So here, actually,
In blue, no, but those in red are very big.
so there's two things. So in the model, we also have some natural.
But they were very small. Actually.
Well, I didn't show them, but they are small.
You
Yeah.
showed it, but later you showed something with the model.
Yeah, but I showed the mean only.
This one.
So this is the mean. We do have some noise around this,
It's not the-
but no, but we don't. I mean, with the model, we do not recover the differences in the variance. So what we think it is, this is a result I didn't show. So this variance is affected a lot by the porosity in the experiment. So basically, when these cells aggregate, they aggregate on some agar, some gel. So by changing the concentration of the gel, you change the porosity properties, and that changes the variance.
And you cannot control the
You can control the gel, but I mean, actually, if you do like a super diluted gel, you may get like a very small. You can change the variance in this experimental result, but we think that there is some natural variance that you cannot even control with the gel, because if you allow, so basically the whole point is that these cells, we think these cells synchronize much faster than the other ones. So if you put this strain in a tube and you mix them for a long time and you then plate them, you have like almost the same variance that you have here. So if you force that synchronization externally, you can recover this low variance.
When you talk about the inheritance,
Mm-hmm.
I'm not sure what you mean. I mean, you mean that individual cells can inherit from their parents, from the cell they were born, this factor alpha, or there is each species has the same alpha?
So by inheritance, I mean that for a population of a given strain, you can expect that to be conserved through different aggregation, so in different aggregation processes. So through different generations, that's inherited from. So basically, the aggregation behavior is
To add to this question, since most of us are physicists,
yeah,
could you be a little more specific on the model?
yeah, but that's not going back on that question, no? It's a different question. It's, no, he was.
I did not understand the model. You just mentioned that there were particles with a random walk.
Okay.
Without a random walk.
Is the?
Because I mean,
I love this question. That's why I come every year.
each bacterium has a different alpha, or all the bacteria have the same alpha?
All the
All the bacteria have the same strain.
have the same parameters. Yeah.
It would be more natural to consider different bacteria have different alphas. I get alpha is probability of aggregation, right?
Yeah.
Has to be with-
But
Without being natural that different different material have different probability.
So the thing is,
Yeah, so they're inherit from one to the next.
so this is, but the alpha is like a population level parameter. So it's basically the, if you want to say is the probability of a cell from that strain to become aggregated cell. Yeah.
Given the density.
Sorry?
Given the density, the modular density, global density of other cells.
So in that case, we're assuming that this is well mixed. So it's depending on the total density.
I thought it was basic that as cells move, the local density decreases.
Yeah, but
And then the probability of becoming.
so the model with the alpha is basically we have a population. So for the model with alphas, we start with a population of cells with different strains. So we have I from 1 to ns, different strains, and then they are competing for some resource. X is the population size. Xi is the number of cells of strain I. Yeah. And once they consume the resources is when we have the population partitioning, basically. So we have, it's the same because I have system size 1. So what we do have now is alpha. You agree? We'll have X alpha becoming aggregated cells and 1 minus alpha X become non-aggregated cells. Okay?
But alpha depends on I.
And alpha, well, if you want, sorry. Alpha is the label of each strain. And then basically we have that for that. No, I have one strain, but each strain has two phenotypes, have aggregated cells and non-aggregated cells. So if I initially have 10 strains competing for the resources, then at the end once they consume the resources, I have 20 phenotypes because I have aggregated cells belonging to strain 1, non-aggregated cells belonging to strain 1, aggregated cells so this is the population partitioning, and then what I have is that when there is no food, so then once I have the partition, and they all die exponentially with different rates, and the rate of the non-aggregated is much larger than the one of the aggregated cells. And then once I have the end of the starvation period, then I go back to here. I reset R and I go back to here. And the length of this time is an stochastic variable.
You mentioned
Yeah, because that's why I said that first we have this model and then we have a special model. I can show you now the random walk and I can solve it for you.
Well, I think that
Because I
in
see.
details maybe we can andamos puestos. No, it's wrong.
One of the key factors to explain the loners, or this good explanation of the loners, is that they respond with different. One responds faster and the other responds later. Is this is this a second trait besides alpha, isn't it?
Yeah, but no, because that's we assume that every strain has the same. So all the loners from different strains respond in the same way, and all the germination time for the spores is the same for all the strains, basically.
But the ones, some of them do before and some of them later.
So basically, once the resources are recovered, we have that this population of aggregated cells starts reproducing with some delay, basically. That's why I didn't go into all the details.
So the delay is a stochastic variable? Or
It's a parameter for the same for all the populations, for all the strains. You can assume that there are differences, and then if there are differences, then the diversity increases even more.
No, but if all of them start producing at the same time, all of them will migrate at the same time, and then there will be no loners. So there should be differences in the...
No, because when they germinate and they have resources, and they consume those resources. And then once they consume the resources, is when they respond stochastically.
Okay, so it's because the different density in different places depletes at different
Yeah.
rates, and then some of them will become start before, and then they will start to...
No. Even if you assume that they all starve at the same time, the point is that the response to that aggregation stimuli, to those aggregation stimuli, is a stochastic process. It's a stochastic it happens with some probability.
Model with a stochastic process.
Yeah. So then it doesn't happen that the resources are depleted everywhere at the same time. You may have at some point migration and at other points non-migration, and then where you have migration, you are decreasing the population of cells, and you have some cells that get stuck there.
So the ones that get activated the latest find a low density and do not move. Those
Yeah.
are the loners. So the loners are the ones that activate later.
Yeah. So basically you have.
And that's not the trait. That's stochastic.
So this is one model, right? The thing is that the model, when we explicitly measure model the aggregation process to see where these loners come from, what we have is that the cells have some response rate. They signal with some intensity, and they have some sensitivity to that signal. So they are labeled by 3 different traits that in principle different strains may differ in any of these 3 traits. And we assume that for every cell in each strain, these 3 traits are identical. And then at the end, the loners, so basically the loners are the result of asynchrony between the time that cells need to migrate and the time that cells need to respond. It's an asynchrony between 2 time scales.
At some point you say that the cells need to use a signal molecule to interact. So and you said that there is some threshold. I suppose that it's like a multicellular mechanism.
Yeah.
Okay. Well, at the end you say that the next steps try to understand from a free-radar perspective. So I suppose that you are going to study like cheaters and operators inside this framework. So do you have some insights from what could be the operator and the cheater?
So
you know if the loners produce the signal molecule?
They do, yeah. Yeah, the idea of cooperation and non-cooperation comes actually after. So once the multicellular structure is formed, there are experiments showing that there are some strains that tend to be more present in the spores than in the stock. And the ones in the stock die. So the idea of this imperfect synchronization is that because this cheating happens in the multicellular structure, the cells that do not participate in the multicellular structure cannot be infected by the non-cooperators. So even when the cooperators are very frequent in the fruiting body and they kill the multicellular phase, you always have some reservoir of cells away from the multicellular structure that will be able to recover the multicellular structure afterwards. That's the idea. Es que yo la mala educación no... Sí, lo que pasa es que si cuando he dado esta, incluso en físicos me he metido ahí, no le gusta a nadie. Pero vamos, yo una persona que desde el minuto 2 se pone a dormir ahí, de hecho te juro que si llego a